Hey guys, today we're going on a tour of Oregon State University's Plant Biotechnology Lab. I'm Mary, let's get into it. Okay, I'm joined with one of our grad research assistants here. Can you please tell us your name and your position?
Hi, I'm Zach. I'm a PhD student here at the Plant Biotech Lab.
Very cool. What does a normal day look like for you working here?
I get into lab and I get to work with a lot of bacteria that naturally are able to transform plants. We work with transforming these plants with these various bacteria and doing a lot of molecular cloning associated with that.
Cool. Can you tell us about what clonal species are and how your work is improving their genetic transformation?
Sure, clonal species is a way of propagating a certain genotype, which we desire is seen as an elite genotype, in order to sort of keep its genetics the same over multiple generations. So our work is looking to improve the efficiency of genetic transformation. So with these elite cultivars that have specific genotypes, which are productive in different areas or for different reasons, we're looking to improve our ability to transform them using a diverse set of tools, which we're screening.
What is your favorite part about what you do?
I love how I get to come in and every day is a little different. The progression of the project and seeing how various strains interact differently with these sort of elite species really is interesting to me, and sort of teasing out why some things are better and helping to create tools that the community can use to further their research efforts.
How'd you get into this?
That's a good question. I got into doing this because I was initially an agriculture science major as an undergrad, and I had some really good mentors in my life who sort of saw the potential and pushed me to see the potential in genetic engineering as a means of precise and rapid crop improvement.
What are you interested in doing after you get your PhD?
I hope to continue doing plant transformation research, whether that's within academia or in an industry company or in the government sector. My hope is to continue doing the research that I'm really passionate about.
What do you wish people knew or understood about biotechnology?
We shouldn't be scared of biotech and the advances it presents. We're heavily already reliant on it, and we're able to be so productive in our agriculture systems because of the advancements we've made. So it's not to be feared.
Okay, ending on a high note, do you have any fun facts about biotechnology or your work?
Any fun facts? I got one fun one. We've been developing an ornamental variety that's a transgenic poplar, and it has purple leaves.
Cool. Do you mind showing us around some of the labs?
Sure, let's go check it out. All right, so this is where we keep all of our germplasm. So these are different species and genotypes of trees that we work with in the lab. So ranging from different genotypes and species of populus, like the model species Trichocarpa, some of these guys, as well as different species of eucalyptus, some different hops, and strawberries as well. So we have different experiments where some of these are transformed, and we also have some where they're still natural, and we use them as stocks for different experiments.
How long have they been growing?
So they go for many different generations. So we have on the top marked, so some of these where it says like F22, they've been repropagated 22 different times. So it's the same genetic makeup as the original tree, but it's just been continued in these sterile conditions over time and just refreshed in new media.
What are these?
So these are some genotypes of eucalyptus. We work with various different companies around the world, from Chile to South Africa, and then here in the US, that are interested in eucalyptus, both for wood pulp production, but also for different aromatic purposes, like essential oils and things like that.
Very cool.
So eucalyptus is a pretty difficult species to transform. So then this is just some eucalyptus with these red roots. And this is a gene from essentially a different species that we can incorporate in order to quantify whether or not we have transformed the plant's genes. So some GMO roots there. Like companies will use transformation to make like better plants and crops. We're essentially interested just in tools to be better able to do that. So different species that you can't transform easily, how can we transform them more efficiently, essentially?
Okay, like
Yeah.
quicker or
Quicker,
better.
better, more rate, higher rate of transformation, so more of them. Instead of transforming 100 trees, you only have to do a few, stuff like that, just to make it easier for both the research and like the development side of things. So. Okay. So these are some of the growth chambers. Essentially, after we do some experiments, we transform the plants and we put them on specialized media where we're able to see how good and bad different treatments are at transforming them. So obviously some turn out pretty well, while others are not as efficient. And we're trying to understand how we can take best advantage of some of the best treatments for transforming plants to improve our abilities to do so. Yeah, some of these here, if you already caught the pink aggro, we can use color markers. They're fun just because they're pink. That's essentially it. We can use these color markers in order to see whether or not we have successfully cloned in various constructs we use for plant transformation. And we also use them to disarm some of these natural genetic engineers in order to create new tools for transforming plants. So, yeah, they're fun because they're pink. I'm gonna turn on the light just so they can like work here. Okay. So this is our microscope room. This is where we're able to image some of our experiments and see whether or not some of the treatments we expose the plants to are good or not. So this is an example here with our microscope, and then one of the successful experiments showing a green reporter protein indicating some transgenic tissue development. I don't know. Most of my life is pipetting, so we could do that. I have some pipettes over here that I've already moved over to our. This is what our real lab space is, but we finally today got the go ahead to move back most of our equipment over here. So, hi Carter, are you pipetting things by any chance?
Yes, I am.
Okay, can we get a shot of you pipetting something?
Of course.
But right now he's prepping some plasmids, so we clone using E. coli in order to produce a lot of these circular pieces of DNA that we use downstream in plant transformation. And essentially, he's pulling out all that DNA from the E. coli so that we can work with it. Oh, and this is not hazardous E. coli, by the way, just so you're aware. I know it's like the headline is like E. coli found in something. E. coli are natural resident bacteria of all of our guts. So most of them are not bad.
Here I go using today's band pipette that doesn't palpitate.
But yeah, essentially it's just like you swab a little bacteria and you kind of streak like this, sterilize it, turn, you keep streaking. We do it fairly commonly as a means of getting single colonies, essentially. So it just means that all the bacteria, a little speck will all be the same genotype. Some of these are terrible.
Yeah, get the contaminant.
Yeah.
Wait a minute. Wait.
Perfect. All these are good. Okay.